Our lab researches how to take small peptides and proteins and turn them into possible drugs, where we take bacteria and we use them to engineer a very large polymers that can then be attached to those small peptides. And many people are familiar with a variety of like peptide drugs, like GLP-1. Um, it, it's always a challenge to figure out how to make those so they're injected infrequently. These are some kinds of drugs that need to be injected, need to be brought into the skin, can't be taken orally, but we don't want to take them all the time. And so we need an approach to release them over maybe a month or 2 months. So our particular technology that we work on is it gives us a strategy to really stretch out the length between those doses. A challenge of working with small molecule drugs is because they're so small, they often don't have a lot of surface area to interact with whatever target they're designed against. And so that comes with a lot of non-specific side effects. And so by being able to use proteins, because they're much larger than small molecules, they actually physically have more area that can interact with these targets. And so that makes them more and more specific. We're working on therapies in a variety of different contexts. We have projects related to cancer, including breast cancer, lymphoma, and leukemia. We're also working on autoimmune disorders and disorders of the retina. I was trained as a chemical engineer. I love phase behavior. I love thermodynamics. I love pharmacokinetics. I like math. And I am able to now use these kinds of mathematical tools that are used for engineering in trying to develop these as drugs that could work better in people. And technologies like this will make better drugs because they can be more specific. They'll also be delivered for longer periods of time at constant levels. And I also am highly motivated by the idea of helping patients one day directly with the therapeutics that we're producing.